All right. So with that, we're going to start the next session on AIRR-seq data addressing human health and disease. So our first speaker is Rachael Bashford-Rogers, who leads a group in the University of Oxford developing experimental and computational methods in B and T cell receptor sequencing and single cell transcriptomics. Her research focuses on determining the development, regulation, and function of B and T cell populations in health and immunological diseases, disease susceptibility, and the effect of therapy. And we're very happy to have her here to start us off.
Hello, it's great to be here in Genoa, and thanks to the organisers for inviting me to speak here. So we have a lot of B cells in our peripheral blood. So approximately, to put this into scale, we have approximately 3 million ants in this ant's nest, and we have approximately 10 times that number of B cells in our peripheral blood and many more in our lymph nodes and tissues. And each one of these can produce a unique B cell or B cell receptor or antibody sequence. And these elicit immune responses. So we've heard over the last few days how antibody or B cell receptor sequences are generated through VDJ recombination. This is not my slide. Good to go? Okay. So upon VDJ recombination, we can get somatic hypermutation and class switch recombination. And this can respectively increase or decrease the affinity to the antigen, and B cells can be selected on that basis. And class switch recombination essentially allows the antibodies to bind different Fc receptors. This is a highly regulated process, and we've basically seen in the last few days that the VDJ recombination is like a molecular barcode for each B cell clone. And we developed methods, along with many of you guys, to essentially take blood, tissue, or single cells to perform amplification on these and sequencing to try and understand the repertoire, what it's doing over time, and many different immunological questions to lead us on to how we understand health and disease and how to treat diseases better. And we can also do a similar thing with the T cell receptor, but I'll really focus on B cells in this talk. So what we have is a system where we can take RNA from a sample of blood, tissue, or single cells, and we can perform a single RT-PCR where we can essentially get a total B cell receptor repertoire. And this is a representation where each dot here represents a unique VDJ sequence. The size of the dot represents a relative frequency that we see in the sequencing data. And edges are created between dots that differ by one nucleotide. But what we can do with this data is actually split it up according to isotype. So we can look at an isotype-specific profile as well. And this means for any potential clone in this individual, we can look at the phylogeny of the B cells within that. And this is just an example phylogenetic tree from this individual, where we have, this is the B cell receptor that's closest to the germline. We can see somatic hypermutation, clonal expansion at this point. We can see class switch recombination to a different isotype, shown by the pie chart, and further somatic hypermutation. So we can essentially see B cell diversification, class switching, and proliferation in real time using sequencing data. And we can show this using cell-sorted populations where we get the isotype distributions quite nicely between different B cell populations, as you would expect. For example, the naive B cells are predominantly IgD and M, whereas on the other side, the antigen experienced B cells are mostly class switched. So this has many uses across biology, so ranging from lymphoproliferative disorders, understanding responses to vaccine, infection, malignancy, understanding autoimmunity and inflammation, immunodeficiency, and just understanding the basic biology of B cell development and selection. So here I'll really talk about what are the differences between B cell repertoires between health and immunological diseases. Does genetic predisposition impact on B cell repertoire? And what's the effective therapy that we commonly use on these immunological diseases? So here's really a diagram of what we went to do. So we essentially collected blood samples from a whole range of immune-mediated diseases, some of which are associated with autoreactive B cells and autoantibodies. And this is effectively recruited cohorts. So all of the patients are collected at diagnosis, so minimal pre-existing therapy within these individuals, which is quite important for studies like this. So we performed this isotype specific BCR sequencing to try and understand disease-specific characteristics and effective therapy. And one key point here is that the age of diagnosis is actually very different between these immune-mediated diseases. And we need to account for that in the statistics of the analyses, because we know that the B-cell repertoire changes a lot over the lifetime of an individual. And we account for this in all the comparisons between diseases. So when we look at the isotype usages between the diseases, this is basically a summary heat map of the data. And here we see the red boxes are where it's significantly higher in the disease compared to health, and blue if it's significantly lower in disease compared to health. And as a disease control, we have chronic lymphocytic leukemia, where these are characterized by large IgD-M positive clonal expansions, and that's exactly what we see there. However, in our immune-mediated diseases, we see some strong patterns of isotype usages that are different to health, and some of which are revealing previously unknown immunological discriminants between disease types. And for example, we see elevated IgA in Crohn's disease and lupus, and we can show that this corresponds to an increase in IgA titer in these diseases. And secondly, elevated IgE in EGPA has previously been shown. The interesting thing with this is that we also get increased IgG3 in EGPA as well. And I'll come on to that later. Secondly, we can look at B-cell clonal expansion. So again, these are the network plots that I mentioned previously, where we essentially have each vertex representing a unique VDG sequence. They're colored according to isotype, and we have edges between clonally related B-cell receptors. So here on the scale, we have our healthy individuals that are very diverse, and this is what you'd expect, because we hope that we will basically have a repertoire that can bind any potential pathogen in your environment. On the other hand, we have our chronic lymphocytic leukemia, where we have these large IgD-M clonal expansions. So where do our autoimmune diseases lie on here? So we have a gradient of clonality between diseases. So on the one hand, we have EGPA, Crohn's, and lupus, where we have these large clonal expansions, not to the same extent as a leukemia. And we can see that a lot of these clonal expansions are class switched. And on the other hand, we have ANCA vasculitis, Behçet's and IgA vasculitis that show very similar clonalities to that of health. And we can quantify this. We use two clonality indices that measure different aspects of the repertoire. So we have the clonal expansion index, which basically measures the unevenness of the number of observations of each unique VDJ sequence across the whole repertoire. And this is essentially looking at the differences between essentially the number of VDJs that we see. And we can see that we have some strongly increased clonal expansion indices in EGPA, lupus, and Crohn's. And we split it up, of course, according to isotype as well, to give us an idea about the isotype-specific contributions. One thing to note here is that we've also done this according to a subsampling method that accounts for any differences in isotype frequencies. The second clonality index is a clonal diversification index, which essentially looks at the unevenness of the number of unique VDJs per clone. So this is irrespective of the number of RNA molecules that we pick up per unique VDJ. And it's essentially a measure of somatic hypermutation across the whole population of cells. And again, we get a different but also interesting pattern of clonal diversification indices across the different diseases, specifically with EGPA, lupus, and Crohn's, again, being more clonally expanded. So this reveals that clonality differs significantly between the immune-mediated diseases, and this stands even after correction for age. It shows that you can have an active autoimmune response. So for example, in ANCA-associated vasculitis, where we have a single autoantigen associated with the disease that doesn't show any global abnormality of the B-cell receptor. And there are other diseases such as Crohn's, EGPA, and lupus that are associated with a very marked increase of clonal expansion and diversification. We can also use the data to look at class switch recombination. So class switch recombination, as we all know, is a deletional DNA process. And this means that we can get some directionality as well into this process, judging from the sequencing data. So we can do this by basically analyzing the sequences that have an identical VDJ sequence, but they have different constant regions. And we can essentially assess the overlap of sequences in a way that is done through subsampling to account for differences in the numbers of each isotype. And it's a directional process, as I mentioned, where we can essentially look at the proportions of switches according to the genomic location of each constant region. In this data, we used a method where we can't quite distinguish between IgA1 and 2 or G1 and 2, which means that we can essentially get a network of class switch recombination between these. And indeed, if we look across the healthy individuals, this corresponds quite nicely with previous studies. So in diseases compared to health, these are the network diagrams that we get. So to explain this, we have each isotype around the edge, and the size of the dot corresponds to the frequency of those isotypes in the repertoire as a mean across that disease group. If it's colored red, it's significantly higher in that disease compared to health, and blue if it's significantly lower. And then we have the arrows that are basically a quantification of the class switch between those isotypes, and again, red if it's higher than health, and blue if it's lower. And we can see these extraordinary patterns between diseases. So for example, in Crohn's, EGPA, and lupus, we can see that there's an elevated class switch level between, for example, a number of different isotypes with IgE. And interestingly, in EGPA, we do see a very strong signal between IgG3 and IgE that corresponds along with the relative isotype frequencies. So this is really the first systematic analysis of isotype switching between immune-mediated diseases, and it reveals disease-specific increases in switching that contributes to the observed isotype profile. We also wanted to ask the question about how IgE B cells are generated and their role in the peripheral blood repertoire. So we see that the majority of IgE in healthy individuals is observed in the plasmablast population, despite it being a relatively low amount. And the problem with looking at IgE positive B cells is that they're particularly rare. So in this study, we can show that circulating IgE B cells are typically from expanded B cell clones, shown here. And this is consistent across all diseases. And we know that in the mouse, IgE B cells are typically generated from precursors of other isotypes and primarily differentiate pretty much immediately to antibody secreting B cells. So we wanted to ask the question, does this occur in humans, and what might the implications be? So during class switch recombination, you can essentially get two daughter cells that may have identical VDJ sequences but different isotypes, as we saw with the class switch recombination, the class switching frequency profile I showed earlier. So to test whether IgE B cells could actually undergo somatic hypermutation, we asked the question of, do we actually see the majority of IgE cells present as another isotype, so the VDJs in these IgE cells present as another isotype, or do we see that they form phylogenetic branches in their clonal trees due to somatic hypermutation? And the answer to this is really through two tests, and the answer is that IgE B cells typically do not undergo somatic hypermutation. So pretty much all of the IgE cells are present with their VDJ as another isotype, and their nearest clonal neighbors, so their clonal relatives, are typically of other isotypes as well. So this suggests that circulating IgE B cells do not show evidence of somatic hypermutation, and it suggests that human IgE B cells arise from clonally diversified memory cells or activated B cells from a precursor isotype, and these are potentially transient within the peripheral blood. And what we can do is generate phylogenetic trees of these IgE-associated clones to try and understand the processes going on here. So this is just one example and representative IgE-associated clone in an EGPA patient. It's actually very similar in the case of any of these autoimmune diseases or in health, where we get multiple class switch events to IgE within the clone. So the IgE is in the light green here. And we can see multiple instances of it popping up. And we can also follow these clones over time. So this is just one EGPA patient. They were treated with immunosuppressive therapy during this time. And we can see that the IgE B cell clones persist over the course of therapy. And the branches, so I colored them according to when they're present at that time point. And we can show that the B cell, the VDJ sequences cluster according to time more than they do according to isotype. And IgE is typically present in distant branches rather than neighboring branches, suggesting that the IgE B cells are actually maintained by non-IgE precursors. And you can still get significant levels of somatic hypermutation during the course of pretty stringent therapy. So next I'll talk a little bit about V-gene usages across individuals. And as we know that we can get this VDJ recombination process, and there's several layers of peripheral and central tolerance that can essentially impact on the V-gene usages. So for example, B cells can be clonally deleted or may undergo secondary rearrangement if they are non-reactive or autoreactive. They may undergo the notion of anergy, so a lack of responsiveness to stimuli, or they may be positively selected in the periphery. So by measuring the V gene usage frequencies, we can get a handle on B cell selection across individuals. And we demonstrate this in a small study looking at the Fc gamma R2b receptor, which is associated, a variant of this is associated with susceptibility to lupus, but less susceptibility to malaria. So most people have a variant of the FcR gamma 2b receptor, which is the non-susceptibility variant for lupus. And we show in a mouse model that this variant essentially is associated with two checkpoints, so central and peripheral checkpoints. So we wanted to show in humans whether this was also the case. So we essentially recruited a number of individuals with, that are homozygous for the non-risk and the risk allele. We flow sorted their PBMCs and performed BCR sequencing. And one readout of tolerance is basically looking at the frequency of V434, which is known to be autoreactive and red blood cell antigen in the unmutated form. And there's a specific motif that's associated with this. And indeed, in our lupus patients, we can see that this is elevated in the naive and antigen experience repertoire. And also in our high-risk FcγRIIb healthy individuals, we can see that's also elevated as well. So this is quite consistent. So when we look across autoimmune diseases, we can actually map out the differences in V gene usages. So this is, again, a heat map where red is elevated, significantly elevated compared to health, and blue is significantly lower compared to health. I've ordered the V genes according to their amino acid similarity. And I've also split up the B-cell repertoires according to their naive-like BCRs, so the IgD are mutated, and also their antigen experience, so the mutated or class switch repertoires. We can see these striking patterns of V gene usages between diseases. So for example, the V4 gene families and the V1 families are significantly increased in the majority of these diseases, and V3 is typically reduced. So we can see these significant patterns across diseases, and a lot of the V genes that are associated with an increase are also shown in the literature to be associated with autoreactivity. So we're hoping to follow this up to try and investigate the implications of this. So next I'll talk about the impact of therapy. So for ANCA vasculitis and lupus patients, we actually were able to follow individuals treated with mycophenolate, which we abbreviate to MMF, and rituximab RTX. And these therapies can have a big impact on the number and proportion of each B-cell subset, particularly rituximab, which is an anti-CD20 monoclonal therapy. And we see that the remaining B-cell population is very much depleted and predominantly plasmablasts. So what impact does it have on the B-cell repertoire itself? So MMF essentially changes the B-cell repertoire to typically an IgD B-cell population, whereas rituximab, we see that it's predominantly class switched and increased somatic hypermutation and clonal expansion here. So we can quantify this. So this on the left is basically the isotype usages between the different therapies and between the different diseases. And here on the right, it's the clonal diversity measures. So essentially, we can see significant differences between the different therapies. And MMF essentially reduces the proportion of antigen-experienced B cells, leaving more naive-like B cells within the repertoire, which is consistent with the population changes. Interestingly, though, we do get a relative retention of IgD mutated B cells, which are likely to come from the memory population, compared to class switch, suggesting an implication on the relative efficiency of MMF on different B-cell subsets. On the other hand, rituximab is a B-cell depletion therapy, and despite only a very small number of B cells still remaining in the blood, we get significant B-cell repertoires from them, and they're significantly class switched and clonally expanded. And indeed, we can look at the effect of therapy over time. And the data since last MMF therapy correlates quite strongly with an increase in class switch BCRs and an inverse relationship with rituximab therapy. So the B-cell repertoire does seem to return to normality, according to these measures, after approximately a year. So the nice thing with this patient cohort is that we, for the vasculitis patients, we were actually able to sample individuals at 0, 3, and 12 months. And we were able to look at persistent clones between them. So indeed, we actually see persistent clones present in the majority of individuals, so over 90% of them, irrespective of whether they relapse. And these persistent clones, the nature of them actually differed significantly between the different therapies. So MMF therapy was significantly IgD and M clones, and rituximab was significantly IgA clones that were persistent over time. And indeed, for irrespective of the therapy, we actually see a significant proportion of clones that expanded over the course of treatment, either between 3 and 12 months. So this suggests that despite trying to impose a strong selective pressure, impose immunosuppressant on the B-cell repertoire during therapy, we still see class switch recombination and somatic hypermutation within these clones. And lastly, and possibly the most interesting aspect of this, is that the number of persistent clones actually corresponds to whether a patient becomes autoantibody negative after therapy versus if they remain autoantibody positive. So the number of persistent clones correlates with autoantibody titer, and this may have implications in trying to track or monitor disease. Is the autoantibody levels, at least in ANCA vasculitis, also correlates with disease outcome. So as a summary of this, we show basically a multidimensional way of population characterization using B-cell receptor sequencing. We reveal unexpected differences in isotype clonality and V gene usages between diseases. We reveal differences in the mode of class switch combination between health and disease. We show the impact of different therapies and that there were specific impacts on B-cell repertoire between the therapies. We look at the nature of persistent clones between individuals and between therapies and looking at clonal evolution. So this was mainly work done in Cambridge in my previous position, so I have a lot of people to thank along the way, including the patients and clinicians who provided the samples and many others who I've talked to and worked with. So thank you very much, and any questions?
Great talk. Thank you for the brilliant talk. So about the last part you mentioned about the persistent clones, you say that correlated with the antibody titers. So have you done any analysis on those persistent clones about the phylogenetic trees, their lineages? So did you see any sharing of those persistent clones among different autoimmune diseases or these immune-mediated diseases?
So are you talking about public clones then?
And persistent clones you identify because you said you may have some of the biological implications or
Yeah, so in this data we show that the persistent clones, we show somatic hypermutation within them. So you can basically follow the same clone over time, and we do see additional mutations within them, and we see additional class switch combination. It does show that the therapy itself is not necessarily impacting the repertoire in the way that we might. So there's quite a number of people who think that rituximab reduces the B-cell population to such a small number that really the probability of you getting back that the autoreactive B-cell clones is actually relatively low. What we show here is actually we do retain those clones, and there's a likelihood that they're just going to re-expand when you take away the therapy.
Okay, thank you. So, and again, you just mentioned about public clones. Did you do any analysis on that?
So not in this study. We're beginning to look at some of that, but it's still work in progress.
Yeah, thank you. Great talk. So, I mean, in the diseases you've looked at and treatment with rituximab, you said that it mainly reduces the naive cells and targets and leaves more the memory cells, the B cells.
So I think it retains more of the plasmablasts, and that's what we see in the flow data as well.
So not really the memory B cells, but more the terminally differentiated B cells in a way, or?
Yes, what we do also see is in the retained cells after rituximab is the IgD negative, CD27 negatives.
Okay.
So I think characterizing those cells would be interesting as well.
Yeah, because I mean, I only know from the MS field where people, speculate that the memory B cells are the bad guys, basically, and so rituximab works because you reduce those cells. But if you would say, well, it's more that these cells stay, that would be very interesting, actually, also from a pathobiologically point of view.
Yes, so there are two ways of looking at it. So there's some nice papers showing that IgA plasmablasts are constantly being generated from mucosal surfaces during rituximab therapy, and that probably contributes to that quite a lot. There's also some that suggest that CD20 is downregulated in a subset of cells, and that might actually be some of the memory subsets. I think and also the other thing is that rituximab can impact tissue-resident B cells differently depending on the tissue. So I think there's a lot of exploration to be done there.
Thank you.
That was really cool. For the clonal lineage tree showing the multiple origins of IgE, how exactly are you quantifying the number of origin events?
Sorry?
Of IgE.
The number of IgE cells.
The number of origins of IgE along the clonal lineage tree.
Okay, so in those trees, they were just generated by maximum parsimony, so slightly different from your methods. But essentially, you can look at the nearest neighbors and also the nearest set of neighbors of an IgE cell, and you can see that basically they're an island surrounded by other isotypes. So you can see essentially that there are multiple points of IgE generation within those trees.
Other questions? Maybe I'll ask one. In terms of the therapy, could you show anything particularly about the specificity of those persistent clones, other than that they seem to be there at high frequency?
So that would be lovely to answer. This is done on bulk BCR sequencing. So whilst we could, in theory, generate these antibodies with a pool of random light chains, it's something that we haven't tested yet, but it would be really interesting.
All right, if there's no other questions, let's thank our speaker again.